Welcome, everyone. My name is Anna Leo. I'm a hepatologist and translational researcher from Milan, and it's truly an honor for me to introduce today our speaker, Tim Gritton. He is the co-chair of the Center of Excellence in Immunology and co-director of the National Cancer Program at NIH on liver cancer. He's an expert in gastrointestinal oncology and tumor immunology. Focusing on tumor immunology, he is trying to understand how tumors in the liver interact with the immune system. He received his training in medical oncology and hepatology in Germany and has been performing basic and translational research in tumor immunology for more than 20 years. Today, the title of this translational science seminar series is PSC and Cholangiocarcinoma, where the research stands on the interconnection. Welcome, Tim, please. The floor is yours.
Anna, thank you very much for this very kind introduction. It's always scary when I hear how long I've been doing this. So screen sharing has failed. I wonder why this is. Let me see. Does this work?
Yes, we can see it perfectly.
Okay, so thank you very much again for the kind introduction and the opportunity to talk to you today. The title is PSC and Cholangiocarcinoma, and where the research stands on the interconnection. So what I would like to do is I would like to give you start with some clinical background about immunotherapy of cholangiocarcinoma. So in our own group, we actually were one of the few groups to start very early a study with anti-CTLA-4 in patients with biliary cancer. In this case, the study was based on a prior study that we had done in HCC that was very successful. And we used the same approach also in cholangiocarcinoma and treated patients with anti-CTLA-4 and local regional therapies. The outline of the protocol and the treatment is basically shown on this slide here. And Dr. J led the study and published this in 2019. We treated 20 patients. You can see the characteristics of these patients. The majority of these patients had intrahepatic cholangiocarcinoma, and they all had received prior chemotherapy. Unfortunately, the outcome is really disappointing. You can see here on the left the spider's plot, and we only had really one clear responder in this particular study, which was very different from what we see when we treated patients with HCC. Now, this is a kind of a teaser to where I go towards the second part of my talk. This is the outline and the description of the one case that actually showed an exceptional response. And without going into all the details, I just want to mention that this patient that was actually initially started treatment in 2016, and as of today, the patient is actually right now without any treatment and doesn't show clear signs of tumor, had a very interesting course with multiple issues during the treatment. You can see here there was an abscess and we had to treat with antibiotics. Patient developed severe immune-related adverse events. But the good thing is that this patient did not only develop a PR, but actually is tumor-free now 4 years out of the treatment. A second study that we conducted was now a study where we used anti-PD-1 in combination with chemotherapy, again in patients with advanced biliary tract cancer. You can see the outline here of the treatment. The study is completed. Dr. Monher has submitted the protocol and the results of this protocol. You can see here again, you know, there is obviously maybe a little bit of a sign of efficacy of this treatment, but it's not easy because we combine this with Capecitabine and Oxaliplatin, and it's not really clear what the particular role of the immunotherapy is in this study. So let me summarize. Immunotherapy may work in primary liver cancer, and you may know, or I'm sure you know, it's an approved treatment in primary hepatocellular carcinoma, but in cholangiocarcinoma, we don't really have very good data. And there are so many unanswered questions. And in our group, we're trying to answer those questions using different ways. One of them is in preclinical models. So Dr. Dix, who was a surgical fellow actually in the lab, decided to test this in a mouse model and test an immune checkpoint inhibitor, anti-PD-1, now in combination with anti-CD40. So what is anti-CD40? Well, I'm sure the audience is very much aware of PD-1 and PD-L1 up here, and CTLA-4, which is basically all inhibitory molecules. Now, CD40 is actually an activating agent, and there are antibodies that you can use to engage T cells by activating CD40. I'm just going to show you the end result of this study here using 3 different mouse models. This is with the subcutaneous tumor. You can see that anti-PD-1 treatment in green really didn't have any significant effect. The anti-CD40 in blue shows some efficacy, but then the combination of anti-CD40, PD-1 is clearly more effective. If we inject tumors into the liver and study orthotopic tumors, again, you can see some minimal effect of either anti-PD-1 or agonistic CD40 as single agent, but a very strong effect if we use both drugs together, and finally the same if we use now a genetically induced model. We also tested this in combination with chemotherapy, in this case with gemcitabine and cisplatin. Again, you can see here, there's strong effect of the CD40 and the PD-1, and there's actually also a good effect of the chemotherapy in this mouse model. So now we decided to use mice, inject tumors into these mice, and wait until we really have a good sized tumor. In 10 millimeter diameter is a relatively large tumor for the mouse before we start the treatment. If we treat these mice with chemotherapy, you can see in blue there is a significant reduction in tumor growth for a short period of time, but then you see these blue and the black lines really overlap. And I want to mention that the treatment period was really only during this area that is highlighted here in red, which is about 2 weeks. Interestingly, you can see that despite only treating for such a short time, we have a significant improvement in overall outcome and survival in these mice. This paper is now out in Hepatology in 2021, and the findings from this proposal or from this study form the rationale for a potential future clinical trial that we are currently planning. So now let me switch gears to the microbiome. And remember, I already alluded to this, that we had one case where we actually saw responses. This patient actually showed a response, showed also complications with an infection, and was treated with antibiotics. So a while ago, and I'm just going to summarize the data here, Dr. Ma from my group conducted a study where he showed that if we use a mouse model where mice develop spontaneous liver tumors and treat these mice with antibiotics, we see a significant reduction of liver tumors. Now, this does not only apply to this one model, but it actually applies to several models where we look at the tumor growth of intrahepatic tumors. You can see here black melanoma tumor cells that we inject through the spleen so that they form metastasis. And again, if we treat mice with antibiotics, there is fewer tumors. And another model where tumors grow in the liver, and again, with antibiotics, there is fewer tumors. Now, I've been a medical oncologist for long enough to understand that you can treat tumors with antibiotics. So it was very important, and I was very curious to figure out, what the underlying mechanism is, how that would explain, why these mice treated with antibiotics actually have so much fewer tumors. And again, since this was published in 2018, and I'm just going to summarize here what we believe what is happening is, and that is basically that the commensal bacteria, so the bacteria that we find in the gut, can actually metabolize bile acids. Now, bile acids are under normal conditions synthesized in the liver and secreted into the small intestine. And these microbiota, and there is a specific microbiota species, Clostridium scindens, that can actually metabolize primary bile acids into secondary bile acids. And if we eliminate this by treating these mice with antibiotics, we change this bile acid pool. Now, the important thing is these bile acids that we have in the intestine are actually reabsorbed and go back to the portal vein into the liver. Once they are in the liver, they can actually induce the expression of a certain chemokine, CXCL16, which leads to the accumulation or attraction of so-called NKT cells, which can kill tumor cells. So what we have shown in this particular study here is that by changing the composition of the gut microbiota, we can actually control through a chemokine-mediated pathway the composition of immune cells in the liver and the anti-tumor immunity in the liver. And this is obviously a novel approach that we are very interested in following up. And we are currently testing this in a clinical trial. I'm going to come to this in a second. But we have actually already some clinical data from patients with cholangiocarcinoma where we show that there is a certain correlation between the chemokine expression and the bile acids that are present in the liver, which basically underlie or support our findings that we have made in mice. So I mentioned that we have currently a study where we test this approach, treating individuals with oral vancomycin to see how we can change the gut microbiome, bile acids in the serum immune cells, and conduct correlative studies. So with this introduction now, I would like to come to biliary cancer and how in our studies, where we actually look at biliary cancer and more specifically, how it relates to PSC. So I'm sure you all know the basics about biliary cancer, so I don't want to go into those details, but the risk factors are interesting. So obviously PSC is one of the risk factors. Inflammatory bowel disease and cirrhosis are other well-known risk factors for cholangiocarcinoma. Why is this so interesting? Well, let me first start maybe with PSC. So these are the typical signs that we see for patients with PSC. We know this is a disease of the bile ducts where we have these biliary structures causing cirrhosis, but also different other problems, including cholangitis, jaundice, fever. And this disease is sometimes associated with ulcerative colitis. The pathogenesis is, I would say, partly understood. We know there are certain genetic factors. The environment certainly plays a significant role, but then again, certain bacteria in the liver can cause this, or can initiate this inflammatory process, which ultimately leads to this chronic cholangitis. We know that PSC patients have a high risk to develop cholangiocarcinomas. What I show you here is a very recent meta-analysis from more than 4,500 studies initially that were included to search it. And the outcome here is shown on the right. The outcome mainly, what do these patients with PSC die from? And you can see that the highest risk for these patients is indeed cholangiocarcinoma. And here again, if you look at the different larger studies, all indicating that cholangiocarcinoma is a risk factor for patients with PSC. Now, based on these studies, since we know that PSC is such a risk factor, there is obviously a great need to perform screening and potentially early detection. And one of the avenues to go is to look for methylation markers, and you can do this in different tissues that's shown here. You can get, you can go directly into the bile duct, can get biliary brushes, or you can get bile or blood, perform blood sample analysis. to look for molecular markers, and that's basically currently being done. Right now, from a clinical perspective, what is done is imaging studies every 6 to 12 months with a CA19-9, which is recommended for all patients with PSC, unless they are younger than 20 years. And then in those cases where patients are either symptomatic or have increasing cholestasis or a dominant stricture, we also perform either an ERCP, in most cases an ERCP or an MRCP. Now, let's go back to this interaction between the microbiome, the PSC, and the PSC. So I previously mentioned that patients with PSC can also have underlying liver diseases. And what you can see here is a study from 2016 where investigators looked at the gut microbiome in healthy controls. You can see this here in green. And patients with either PSC or ulcerative colitis, Crohn's disease, or a combination of the two. And basically, what you see here is that the composition of the gut bacteria is very different from the controls. Now, if we go in more detail and look at the different bacteria, you can see this on the right, that again, the healthy control has a very different composition than those patients with PSC, and inflammatory bowel disease, or the patients with inflammatory bowel disease. So this was really one of the reasons why we decided to say, well, maybe, we should actually look at this interaction and the potential role of the gut microbiome in the context of cholangiocarcinoma in more detail. So we decided to use established models for cholangiocarcinoma, as well as established models for murine models for PSC-like disease. The way this works is in mouse models for cholangiocarcinoma is we use two different models where that one model depends on the expression of AKT and Notch, and the second model depending on AKT and YAP, where we can actually inject plasmids into these mice by a technique which is called hydrodynamic tail vein injection. If we inject these plasmids into the tail of these mice, we can get stable transduction of hepatocytes, and the transduction with these oncogenes then basically leads to the development of cholangiocarcinomas. Now, we combined this, I already mentioned this, with two models which kind of mimic different aspects of PSC. One is a genetic mouse model. These are MDR2 knockout mice. You can see here how they have a bile duct that shows signs of inflammation here. And the second model is one where we perform bile duct ligation, again, leading to significant cholangitis and PSC-like lesions in these mice. Now, the first, and I think very interesting observation we made is, if we treat these, or if we induce PSC in these mice, we actually see changes in the gut. So these are either mice that had undergone bile duct ligation or have a genetic change. So these are MDR2 knockout mice, which induces a PSC. But if we look at their gut, we can see clearly signs of inflammation and colitis. So on the top, you see the normal gut microscopic picture with an intact epithelium. And now you see that basically this intact epithelium is completely missing. Severe, the crypts are lost. There is an infiltrate of immune cells, all signs for severe colitis. And if we look at different proteins, this case occludin, which is one of the tight junction proteins, they are very, very sparse. If we take the tissue and look for markers of inflammation, for instance, IL-17, we see that those pro-inflammatory markers are increased in these mice. And if we look at the portal vein blood, so remember, this is basically the blood that drains the content, that drains the blood that is derived from the large and small intestine and goes into the liver, we can actually identify or detect high amounts of 16S RNA, which are derived from bacteria, suggesting that there are indeed bacteria going through the portal vein into the liver. Another experiment is called the fitsidextran uptake, where we feed these mice with little plastic particles that we can identify by flow cytometry. And we see if these mice again have colitis and basically a decreased, what we call leaky gut here, or decreased gut barrier. We see more uptake of these labeled particles in the blood of these mice. And just on the side, if we look at patient samples with cirrhotic liver, we can actually also find signals that bacteria or LPS are present. Now, if we understand, or if we study the immune cells in the liver in more detail, we noticed that mice with PSC have an increase in what we call myeloid-derived suppressor cells. Now, what are these myeloid-derived suppressor cells? Well, if we think about the normal production of blood cells from stem cells, you know that under normal circumstances, one develops granulocytes, macrophages, DCs, monocytes, etc. Now, in the presence of a tumor, tumors can actually release certain cytokines, soluble factors, which basically block the maturation of these immune cells. And instead of developing mature granulocytes, macrophages, etc., what happens is that you will find in these individuals what we call these immature cells. And these are also called myeloid-derived suppressor cells, and they can be described in mice as CD11b positive, GR1 positive cells. Now, we can further identify these cells or divide these cells in two different subsets. So-called granulocytic or polymorphonuclear MDSCs, which have certain markers, or monocytic MDSCs. And what these cells ultimately do is they basically cause immunosuppression, and they suppress other immune cells, effector cells, such as CD8 cells, CD4 cells, regulatory T cells. They can induce regulatory T cells, and so on. So basically, this is a mechanism that the tumor has developed to escape a potent anti-tumor immunity. And so far, it's always been thought that this induction of myeloid-derived suppressor cell is really tumor-dependent, and that tumor-derived cytokines really regulate the accumulation of this cell. So let's go back to our mouse model. I showed you that in the context of PSC, there are more MDSCs. If we now look at the two different subsets that I just described, there is a specific accumulation of so-called polymorphonuclear MDSCs, and we find this in the second PSC model also. Interestingly now, if we treat mice with an antibiotic neomycin, we don't find this accumulation anymore. You can see here the mice that have PSC by them without antibiotic treatment, and then with antibiotic treatment, the number of cells in the liver is drastically reduced. And we can perform so-called FMT studies where we now transfer stool samples from mice that underwent treatment with antibiotics to specifically eliminate bacteria, gram-positive or gram-negative bacteria, depending on the antibiotic that we give to these mice. You can see again that those mice that have been treated with neomycin or have received stool from mice treated with neomycin don't really show this accumulation of myeloid cells in the liver anymore, suggesting that the bacteria or the presence or the absence of the specific bacteria really can regulate the accumulation of these MDSCs in the liver. Now, we looked at these MDSCs in more detail. We looked at certain markers on these cells, and we found out that these cells primarily express, of 94% of these cells express a certain receptor, CXCR2. You can see here the accumulation of CXCR6 positive cells. Now, CXCR2 is a receptor on the cells, and it binds to another chemokine, which is CXCR1, which is actually produced in the liver of mice either with PSC, but also in the mice with colitis. And you can see here, hepatocytes actually produce this chemokines in response to an insult, either by induction of cirrhosis or colitis. We can block this by interfering the CXCR2, CXCR1 pathway, either using antibodies, or through pharmacological inhibition. And last but not least, we can actually target the production of this chemokine and the accumulation of the cells by specifically eliminating a receptor on hepatocytes, which is needed to recognize bacteria or bacterial products, and then to start produce this chemokine CXCL1, which is TLR4. You can see here on the right, MDSCs accumulate in the liver, but if we actually knock out this particular receptor on hepatocytes, there is no longer an accumulation of MDSCs in the liver. So what I've shown you so far is basically liver cirrhosis causes a leaky gut. This in turn leads to a chemokine expression by hepatocytes. The hepatocytes start to express a chemokine, and this whole mechanism is dependent on TLR4. And I showed you that the bacteria that neomycin can block this, suggesting that it was indeed gram-negative bacteria. And TLR4, the receptor on hepatocytes, binds to LPS. So we believe that it's actually the LPS from the gram-negative bacteria that plays a significant role here. Now, what I haven't shown you really is any connection from the PSC and the gut microbiome to the cholangiocarcinoma, and this is shown here. So now we use the same model that I've shown you, that I've described before, where we study the PSC and also induce biliary cholangiocarcinoma in these mice. So if we do this, you can see that those mice that also have PSC develop significantly more tumors. This is in one model, and down here you can see a second model where we basically make the same observation. The same also applies to mice where we induce a colitis, and remember, PSC is associated with colitis. Now, without going into all the details, but what we did here is we basically showed that indeed myeloid-derived suppressor cells can suppress tumor growth in this model. So if we take out the MDSCs, we find actually fewer tumors. If we block this interaction of the different chemokines, which are needed for the MDSC accumulation, we can see more tumors if we overexpress this chemokine, or less tumors if we block this chemokine. And again, by changing the gut microbiome and taking away the gram-negative bacteria, ultimately this will lead again to fewer tumors in these mice. And this mechanism is dependent on the toll-like receptor 4 on hepatocytes. You can see here, if hepatocytes don't express this receptor, we don't find more tumors. And we can partly reverse this if we overexpress this chemokine in the liver. Now, we also looked actually at a gene signature in patients with cholangiocarcinoma, where I mentioned this TLR4 gene to be very important in this aspect before. And you can see here that those patients that actually have this gene signature have a worse outcome than those patients that don't have one, suggesting that this mechanism that we initially described in a model where we combine PSC and cholangiocarcinoma may actually also apply to patients with intrahepatic cholangiocarcinoma without a PSC. We also had the chance to look at a few samples of patients with intrahepatic cholangiocarcinoma and inflammatory bowel disease. And we looked at these myeloid cells in the liver, and it appears that these cells are increased if these patients have an active ulcerative inflammatory bowel disease. We don't find this if these are not present. So let me summarize what we believe is happening in this particular study that was conducted by a former postdoc, Shanwei Zhang, in my group is the following. Under normal circumstances, there is our microbiota and gut epithelium in the liver here. But once there is an insult to the liver, and this can be, for instance, PSC, or it can be cirrhosis, this will actually change the composition of the bacteria in the gut and cause dysbiosis. Remember, I already showed you that this has been presented and identified, described for patients with PSC. Now, this dysbiosis by itself is probably dependent on the change in the bile acids, which is associated with the cirrhosis. But more importantly, it leads to a leaky gut so that bacteria can actually more easily transmigrate through the epithelium into the portal vein. Now, either bacteria or bacterial products make it into the liver, where they induce the expression of a chemokine CXCL1 by hepatocytes. And this is through a TLR4 dependent mechanism. Now, release of this chemokine CXCL1 will cause an accumulation of the mononuclear or granulocytic MDSCs, which in turn can suppress NK cell and other immune cells in the liver, ultimately leading to increased tumor growth. So I think what this study here shows, if you remember when I gave you the background about the MDSCs, is that so far we were always under the assumption that the tumors release cytokines and thereby control the accumulation of MDSCs and which causes an immune evasion mechanism. But what we can show here that even the gut bacteria, in this case, changes induced by dysbiosis or by cirrhosis, can actually also control this accumulation of the NK cell. And this is even in a tumor independent manner. And with this, I would like to thank and close and thank you for listening and the lab members. I think I mentioned the members in the lab that were responsible for the particular studies. And with this, I'm happy to take any questions.
Thank you, Tim. That was very inspiring, really. I don't see any questions in the chat, but I invite all the participants, if they have any questions, to just post something. I will start with a curiosity. As you know, the only therapy that is used nowadays for PSC is ursodeoxycholic acid, which is not widely recommended, and there is no real proof if that's of any benefit for the patients. Actually, high levels of UDCA have been proven to be to promote tumors in PSC patients. Do you have any insights in that? What do you think happens in the gut of those patients we treat with biliary compounds?
Yes, so we never tested this, so I can't give you a straight answer, but I think we all know and should recognize that the gut bacteria, they are in a very narrow equilibrium, and whatever you do, just changing the diet, if you change to a vegetarian diet, you will actually already see a significant change in the composition of the bacteria in a very brief period of time. Treating with ursodeoxychol is certainly going to change something. It would be very surprising. There's many reasons why this is, and I showed you one of the reasons where different compositions of primary to secondary bile acids changes. So I'm pretty sure that you will find changes in the gut bacteria. And then what this ultimately leads to and how this may promote to tumors is, I think that's still open question.
Yeah, of course. That's another thing is that, so I practice in, there's a question. Let's see. Were there any damages on the normal liver tissue after NKT cell increase induced by antibiotics?
That's a very good question. No, there wasn't. We didn't see, the question really is, one of the concerns you always have is kind of autoimmunity. If you talk about tumor immunity, the counterpart is autoimmunity. And we do know that whenever you induce an immune response, which is too strong, would this also cause autoimmunity? And in this case, we didn't see this. We do know that NKT cells can cause significant damage to the liver. There are other marine models where we have seen this in the context of carnivalian A, for instance. But just by treating with antibiotics, we didn't see any of this.
Okay. So there is another question from Marcus Schneider from UPenn. Many thanks for this beautiful talk. Do you think that these mechanisms also explain the higher tumor incidence in PSC patients besides cholangiocarcinoma? Do you think that changes of bile acid composition and microbial bile acid metabolism are important? Maybe there are unifying mechanisms that explain the generally increased tumor risk in these patients.
So that is certainly possible, but what I, what I didn't stress is that the effects that we study are primarily specific to the liver. So this accumulation that we find is really something that applies to the liver, and it's not necessarily something that we also see in other organs. So it's very hard to say, but we also have to be very cautious with this. And these are animal models, and these are animal models where we see a very significant phenotype in a short period of time. Otherwise, we couldn't do these studies. We can. So there may be certainly subtle differences that we don't see, and that in an individual, in a patient with a PSC over time. And when I say over time, this may be a period of 3 or 5 years. Obviously, it's something that we cannot mimic in an animal model, may actually play a role. And it's just, in the model where we over amplify this, we see this, but we would not see this during such a short period.
Okay, and what do you think about genetics? Because, in Southern Europe, like in Italy, we don't see that many cholangiocarcinoma in PSC patients. We see very lower rates than reported in Germany or Scandinavia or even the Mayo Clinic.
Yeah, no, there's no question. So, and that's actually twofold. So there's obviously, enough polymorphism and other genetic reasons why there are differences in certain tumors depending on what population you look at. But at the same time, if you look at the gut microbiome, that's actually also different because that's, again, affected by the diet. Now, I'm not saying that everything is related to the gut bacteria, but, this is certainly, a multifactorial process.
Okay, thank you very much. Another curiosity is, have you studied this stroma? Because even both cholangiocarcinoma and PSC have a very strong fibrotic component. And do you think the microbiome is somehow responsible? There is any connection? Do you have data on that?
No, that's a very interesting question, and we're trying to look at this. It is, it appears to be very similar to pancreatic cancer, actually, where we also see this stroma, and there is some indication that this may be that part of it may be related to inflammation or potential microbial components.
Okay, that's interesting. Looking forward to seeing that. So I don't see any other questions from the audience. I would like to thank you. Oh, someone just thank you very much. Interesting talk. I have a question regarding the models. You are using the TBI model that targets hepatocytes to induce cholangiocarcinoma. Do you think these observations will be reproducible in a genetic model that starts cholangiocarcinoma from cholangiocytes?
Yes, so there is a long discussion about animal models and cholangiocarcinoma, and those who are the experts in the field, obviously, you would first of all, we still don't know how the cholangiocarcinoma, what the starting cell in a human actually is, if it's really the cholangiocyte. So that's number one. Number two is there's very elegant studies from the Zender group in Germany showing that that you can actually change using the same mechanism and changing the environment. You can actually make out of a cholangiocarcinoma an HCC and the other way around. So that's number two. Number three is, I hope I stress this enough, that any animal protocol or model is always has certain limitations. Obviously, yes, we do primarily transduce hepatocytes. I don't think we would see a strong, a very difference if this is, if we were to use a genetic model. And the reason why I'm saying this is that the observation that we actually made is that in many cases, the immune responses in this particular environment in the liver is actually independent of the primary tumor. Very simple, we see very similar mechanisms to metastasis and inhibition of tumor growth of metastasis versus primary tumor, for instance. And that gives me reason to believe that if we were to use a different cholangio model, a genetic model, we would see similar results.
Thank you very much. So I think we can, will you expect your findings applied to HCC as well?
I would expect that you will see similar findings, yes. Because if you have a cirrhosis, remember, the initial observation we made is it's in this PSC model, but we know that cirrhosis by itself can cause dysbiosis. We know from patients that have been studied, that they have a different composition. So while I don't, can't predict that you see exactly the same, but I'm sure there are certainly significant changes in patients with cirrhosis. And last but not least, we know probably even more. We have even better data, human data, suggesting that MDSCs also play a significant role for patients with HCC and outcome.
Yeah, that's very interesting. Right. Thank you very much.
Okay, thank you.
I think we can close it here. I would like to thank the Cholangiocarcinoma Foundation for giving us this opportunity and for really giving the opportunity to everyone to watch the webinar that will be posted in their website. Thank you, Tim.
Thank you very much. Have a good day.
Bye-bye.